Welcome to the class. Today we are going to talk about embryo culture part 1. I'm Dr. Chavi Sharma. So let's talk about what is embryo culture. First of all, where is embryo present? Embryo is the part which is present in a seed, which goes on further development in the whole plantlet, in the whole shoot and then root. Okay, so what is embryo culture? The embryo of different developmental stages formed within the female gametophyte through sexual process and can be isolated from the maternal tissue of ovule and ripe seeds. Isolated embryo then cultured in vitro under aseptic condition and controlled physical conditions in glass vials, or we can say in laboratory conditions, containing nutrient solid media or liquid media to grow directly into plantlets. Okay, so according to Perrick, there are two types of embryo culture. First is mature embryo culture, which is done for seed dormancy, and another one is immature embryo culture, or we can also call it as embryo rescue, which is done to avoid embryo abortion. Embryo abortion can be due to so many causes, so many physical environmental causes, which we are going to discuss next. So you can see here the picture. The properly developed embryo, which is mature embryo, and the embryo which is not properly developed is an immature embryo. Okay, so in the seed, in the germinating seed, we can make a cut from the middle of the seed from vertical to downward side, and we can isolate it out. Okay, now why we are doing embryo culture in vitro? If it is already developing in fields and naturally it is developing, then what is the reason why we are developing the embryo in vitro cultures or in laboratory conditions? So first of all, when the embryo remain dormant for a long period, suppose if an embryo is not able to develop in full fruit or in full seed or then in full plant, then at that time we call an embryo is in dormant stage. Okay, so at that time we took that embryo and we uh, culture it in laboratory conditions. Okay, now another one is low survival of embryos in vivo. In vivo is natural condition, right? So if the embryo is of low survival, why it can be of low survival? Maybe if we are making hybrids of different species, either it is interspecific or intergeneric, if we are making hybrids, so in that case it is possible the embryo will survive less. Okay, so the probability of survival of embryo is low at that time. So in natural condition, when the probability is low, we isolate those hybrid embryos and we culture them in laboratories or in vitro. Okay, now third one is for converting sterile seeds to viable seeds. If the seed is sterile, it is dormant or it is not able to develop shoot or root. Okay, or we can say plumule, it is not developing more. Then at that time we can isolate the embryo and we can make a seed of it, and then we help the plant to grow in the laboratory condition. Okay, so what is the two most important aspects of embryo culture? First is composition of culture media. Composition of culture media is very important for development of embryo, because the embryo is going to develop in a plant, you know. The further stages of that is going to be developed in a plant. So we have to provide the whole nutrient components which is used by a plant to survive, or which are used for its growth, which are taken up by soil. Okay, so we are providing those nutrients in the media. So it is very important to take a look what we are making media for, and what kind of plant we are growing, in what kind of environment the plant grows, what is the special need of that plant, either it needs some sulfur content, or what kind of content it is needed, plant is needing more. Okay, so we will just make the culture media accordingly. So the first important aspect is composition of culture media. Second one is excision of the embryo. How you are isolating the embryo from the seed or the female part of the plant. Because if the embryo is damaged during the isolation process, then the whole process of in vitro culture will be diminished, or the plant growth will not take place. So you have to take care of the isolation process or excision process of embryo. Okay, another one is embryo culture can be grown on a simple inorganic media which is supplemented with energy source, usually sucrose. See, growth of plant needs a lot of energy, okay, because it is using that energy for photosynthesis. Synthesis, we are giving it artificially sucrose, you know. So we are, no, giving it a sufficient quantity of sucrose so that the energy can be utilized and it can be converted into the growth molecules. Okay, so the plant growth needs simple inorganic medium, and we just fill it with the sucrose energy. Okay, okay, now the composition of the medium. What is the composition of medium? A basic composition I'm telling you. Okay, it can vary according to the species also. Okay, if some plant species needs a special condition of survival or a special condition of nutrients, then we can change the composition of media accordingly. Okay, so the basic composition is inorganic constituents of ms media, B5 or White's media are adequate. MS, B5 and White's media are available in market. So you can bring them and you can just make the media, and you start growing the plant in that. You just put your explant in that plant in aseptic conditions, of course. Okay, so the list which is used in MS, B5 and White's media is enough. Next, we are adding, as I told you before, we are adding sucrose as an energy source. Next one is ammonium nitrate is the preferable used as a source of energy, source of nitrogen, sorry. Then casein hydrosilate, which is rich in various kind of amino acids. Amino acids are used to make proteins for the plants. Okay, so certain natural plant extracts with embryo factors are used. For example, liquid endosperm of coconut milk. Liquid endosperm of coconut milk itself contains a lot of nutrients which can give a natural environment to the embryo. Okay, so sometimes to fulfill the limiting factors of the composing media, we add some coconut milk of endosperm and further natural plant extracts. Okay, the pH range of the media should be between 5 to 7.5, and the incubating temperature should be 24 to 26 degrees centigrade is ideal. Okay, better growth of embryo is observed in darkness, which are then transferred to light for germination. In the initial weeks, यानी कि first or second week of explant growth, explant is embryo here. Okay, so when we grow embryo in the media, so first to second week it needs a dark environment. Okay, after the second week, if you are providing light to it, then it will be good for germination. Yeah, because embryo's tendency is to stay in the dark, that is why. So this is the list of components of media. Here is the growth media. We are describing B5 media salts. Okay, so this is germinating media and this is growth medium. See, seed, if we are taking seed as an explant, then we want it to germinate. Okay, so we are using germination media. And if we are using an explant like a callus, a leaf, then we can use a growth medium. Okay, because we just want it to be growing. Okay, so this is the difference between the growth media and the germinating media. So what we are using: vitamins, nicotinic acid, thiamine plus with HCl, pyridoxine HCl, myoinositol, and amino acids: L-alanine, glutamine, L-serine, tryptophan, cysteine, NAA, agar, sucrose. Okay, so another part which I called as in vitro culture of immature embryos is called as embryo rescue. So what is embryo rescue? Embryo rescue involves the culture of immature embryos to rescue them from unripe or hybrid seeds which fails to germinate. When the hybrid seeds or unripe seeds are not able to germinate properly because of maybe environmental conditions or interspecific or intergeneric conditions, then we use this embryo rescue technique where we just isolate the immature embryo from it and we plant it in vitro culture, okay, in laboratory conditions. Okay, so what are the applications? Why we are doing this whole lot things? So what are the applications of embryo culture? Prevention of embryo abortion in white crosses. White crosses is interspecific and intergeneric, what I said before. Next, production of haploids. Okay. Then third one is overcoming seed dormancy. When the seed is dormant, it is not going to germinate in any case, then we just take out the embryo from it and we do in vitro culture. Okay. Shortening of breeding cycle. See, normally when seed germinates and the plant grow up to its life, when it can start flowering, it's a long process. Okay. Sometimes it's 6 months, sometimes it's annual plant, or sometimes it's a seasonal plant. Okay, so it's a long time. So if we want to shorten that time, then we can use in vitro embryo culture. Okay. Next one is clonal propagation. Okay,